Okay, great. Ooh, that light is pretty intense. Well, thank you very much, Angela, for the introduction. I'd like to thank the organizers and the sponsors for what appears to be a very exciting and informative conference. What I'd like to do this afternoon is present you with a strategy that we think will eventually be the primary way to manage cancer. And I'd like to give background information first on where this concept of press pulse originally came from, and then I'd like to briefly go over what we view as the cancer code, and I'll explain that, and then how we apply the press pulse strategy to a particular type of cancer called glioblastoma, and then I'm going to show an overview of how the strategy can be applied for almost every cancer. Let's see. All right, where did this idea of press pulse come from? All right, this is actually derived from the field of paleobiology. It was considered a mechanism responsible for the mass extinction of organisms during prior evolutionary epochs. So press disturbances produced a chronic stress on populations, while the pulse disturbance produced an acute stress on populations. But it was only when both of these stresses coincided was there mass extinction of organisms. So you can see the connection. We take this concept and now we apply it to the cancer problem because the goal is to resolve the disease, all right? We want to exterminate cancer cells, but we want to do it in such a way that we don't harm the body or the organ that we're working with. So before we can understand how to apply a press pulse therapeutic strategy for the management of cancer, we have to understand what this disease is. And I put this up here as the cancer code, all right? I put it up there because in my neck of the woods in Boston, Dana-Farber comes on the radio and the TV telling me or us that they have the cancer code, which is based on the gene theory of cancer, all right? And we know from a lot of work, the gene theory is now under attack. And as Travis Christofferson would say, it's becoming a dead theory. So what is cancer? It's a mitochondrial metabolic disease where fermentation gradually replaces respiration for cellular energy. This is Warburg's central theory. Glucose and glutamine are the major fermentable fuels for driving cancer growth. All right, the cancer cell can ferment other things in the microenvironment. The issue is there's not enough of those other things to drive the beast. Ketone bodies can replace glucose in normal cells, but not in tumor cells. This is because the tumor cell has defective oxidative phosphorylation. Tumor cells are abnormal. The mitochondria in tumor cells are abnormal. Ketones cannot be fermented. Ketones must be respired. Reactive oxygen production is greater in tumor cells than in normal cells. Why that is because why the ROS don't kill the tumor cells? We've seen that. We had this debate the other day about the ROS, the reactive oxygen species. These cells are making more ROS, but they're not dying. They're not dying because they're fermenting glucose and glutamine, which produces tremendous antioxidant capacity in these cells. So even though they're making more ROS, they're actually protecting themselves by fermenting these particular fuels. Cancer mutations arise as an effect and are not the cause of destabilized energy metabolism. This is very important because the somatic mutation theory is chasing all these gene mutations, right? These are effects, they're not the cause, they're red herrings. The field is wasting massive amount of time chasing those things. It's the metabolism that's causing those things, it's a randomness. Cancer mutations make tumor cells more vulnerable to energy stress than normal cells. How do you think, here's a cell, full of mutations, broken chromosomes. You think that cell is going to be tough when put under energy stress? Every cell in our body earned the right to be here. We have an evolutionary history of survival, extreme stress in the environment. And then all of a sudden we're claiming that these cells with all kinds of broken chromosomes and mutations are going to be more fit than the cells that we have that evolved? Wrong. Those cells are weak and we just have to know how to exploit their weaknesses. Glutamine-addicted neoplastic macrophage or fusion hybrid is the origin of the metastatic cancer cell. These cells naturally have the capacity to enter and exit tissues, live in the bloodstream, suppress the immune system, all right? This is the cell that we're dealing with. These cells evolved to live in hypoxic environments. They're tough cells to kill. The press pulse therapeutic strategy is going to possibly manage and possibly resolve the disease, as I'll explain to you. And I'm going to demonstrate this with glioblastoma multiforme. Among the most aggressive of all primary tumors, poor prognosis, we know this, no effective therapies, composed of multiple cell types, consequently the term multiforme, because there's different forms of the cells. There are stem cells and neoplastic mesenchymal microglia in this gemisch of cells. The stem cells will grow like crazy, but they are not the cells that invade throughout the brain. They cause mass displacement of tissue. It's the neoplastic mesenchymal microglia that invade through the brain, and they invade using these secondary structures of Shearer, which were described many years ago by Professor Shearer. There are anatomical regions in the brain, and the tumor cells will use these anatomical regions to spread throughout the brain. Consequently, it becomes almost impossible to cure GBM with surgery alone. And the other characteristic of this tumor is they metastasize. When GBM cells escape from the brain and get into the body, they spread like crazy. Powerful metastatic disease. Unfortunately, it doesn't happen very often, but when it does, it's devastating. Now, this is a cross-section through the brain of a person who died from GBM, and you can see the large necrotic area, cystic area. You can also see the compression of the ventricle. Most of the patients who die from this disease die from intracranial pressure. So, and the issue, the normal appearing brain area, let's see if I can get that here. Okay, they're laced with tumors, so you just can't see them. They're not that obvious. But they get to the other side of the brain. They use blood vessels. All right? They use these blood vessels as railroad tracks and other anatomical regions to spread throughout the brain. That's why you can't cure these things with surgery, because these tumor cells are already spread throughout the brain. And they use blood vessels as one of the means to do this. They go across the Virchow-Robin space on the railroad tracks, and immunotherapies can't get to these guys. They're on the opposite side of the brain. That's why that's not going to work. Now, the other thing is the mitochondria are damaged in GBM. All right, look at this is a normal mitochondria. Stripes, those stripes are cristae. They contain the proteins and the lipids that are responsible for electron transport, the oxidative phosphorylation capacity of the cell. This is a mitochondrion of a GBM, a large matrix, small amount of cristae, crystallysis. Structure dictates function. If the very structures needed for oxidative phosphorylation are damaged or missing, the cell can't get energy from oxidative phosphorylation. It's just that simple. So how is the cell going to survive? It has to ferment. What does it ferment? Glucose and glutamine. Now, this is another work from Ruth Dighton's group, where she looked at did a big proteomic analysis in GBM, and they were looking at normal mitochondria and abnormal. So if you look at normal mitochondria, most of the normal mitochondria are found in normal brain sections, and not in the GBM. Have very low normal mitochondria. Just the opposite, abnormal mitochondria are enriched in the GBM and very, very few in normal brain regions. And you can see these dense cristae? Those are normal-looking mitochondria. Here's in the GBM, you see all the cristae. These cells can't respire. How people say these cells can respire makes no sense to me. The anatomical structures are here. All people have to do is look at the data. Now, what's driving this disease? What's driving the beast? It's glucose and glutamine. They're fermenting. So glucose comes in through the glucose transporters, and Christos just gave us some instruction on this. Glucose 6-phosphate, Embden-Meyerhoff-Parnes pathway to produce lactic acid. Now, there's some debate, might not be as much energy coming from substrate level phosphorylation and glycolysis, but that's probably in some tumors, maybe more in others. Lactic acid is dumped out, causes acidification of the microenvironment, contributing to wound healing and all the angiogenic factors that you see. Also, glucose 6 can go down the pentose phosphate pathway, producing ribose 5-phosphate, a precursor for nucleotide synthesis. It also produces NADPH, a substrate for glutathione. And as I said, the glutathione is an antioxidant system. It's one of the main antioxidant systems. So we're keeping our antioxidant levels high. At the same time, the glutamine is coming into these cells. It's split through the glutaminase to glutamate and ammonia. The ammonia from the amide nitrogen is used as a precursor for nucleotide synthesis. So some of the glutamate is pumped back out into the microenvironment. That we know that glutamate in the microenvironment allows these cells to become even more aggressive because the cells are going to be, excitotoxic death is going to contribute from all this glutamate dumping. And as we just heard from Christos, the glutamate can drive substrate level phosphorylation in the TCA cycle to very high levels. So these cells are getting their energy, their metabolites, and also the glutamate is part of the glutathione complex. So glutamine and glucose are building a very powerful antioxidant system, building all the metabolites that you need to grow the beast. All right? How many therapies are we using to stop this? How many of the drug therapies, the immunotherapies, all the things that we're using to stop this, are we doing this? I don't know of any that are combining both together simultaneously. Now, what do we do for the standard of care? All right? So we take somebody diagnosed with a GBM. Before they even have the opportunity to really understand what's going on, we do surgical debulking. So we take the surgery out. It's like hitting a beehive in the middle of the day. The bees come flying out. All right? So you create a wound, and that wound is now going to initiate these tumor cells to start spreading. As soon as the patient has the opportunity, we start irradiating that person with high-dose radiation, creating necrotic environment, breaking apart the glutamine-glutamate cycle, freeing up glutamine that goes into the tumor cells to drive their fermentation metabolism. Okay, we create a necrotic environment, excitotoxic death. The head begins to swell. We have edema. How do we stop edema? We throw in massive doses of steroids, dexamethasone, which elevates blood sugar. Blood sugar, the other component for driving the beast. So the standard of care is creating environment to make sure that tumor, or to facilitate that tumor's recurrence. All right? So, and these things are, many of them are affected with human cytomegalovirus, which is a supercharger for driving glucose and glutamine into those tumor cells. The worst condition. And when Adrian said the other day, GBM is a perfect storm, the perfect storm is created by the standard of care. And look at the results. Here we're irradiating these poor people. Nobody survived. 0 survivors. This is from one of the Stoop protocols. And then we use radiation and temozolomide. We get a few extra survivors. But if you look at it, 6 out of 500, 1.1% of the people treated. What do you expect from a therapy that's creating the perfect environment for the fermentation of these cells? You would not expect anything different from this. Make matters worse, you throw in this toxic alkylating agent, temozolomide, which increases driver mutation. It makes no sense at all, because these people have a little bit of an increase in progression-free survival from a drug that increases driver mutations. What does that mean? Nonsense. This whole thing about the gene theory is just such nonsense. Now, I put this up here. This is Remembering Brittany Maynard. Tragic situation. You may have read about her in People magazine and all these kinds of things. I mean, here she was right after she got married. She was operated for by a low grade, and within a few weeks it turned into a high-grade tumor, all right, GBM, unbelievable. So she says she's taken the standard of care. She says, I don't want to go through this. So you can see, look at her here. That's moon face. That's from high-dose steroids that she's receiving. That's the result of that. She says, I don't want to go through this anymore, right? So she lives in California, goes to Oregon, right to die. So she dies with dignity on November 1st, within less than a year, right? So the question is, in this article, it's all the morality of right to die discussions, nothing about the failure of the standard of care. If the standard of care weren't so bad, maybe she wouldn't have to do this. Now here's Pablo Kelly, made an alternative choice, all right? Recent paper, this came out last summer, July 15, 2016, in the English paper Herald Express. Diagnosed with GBM, he's approximately the same age as Brittany, diagnosed with GBM in August 2014. He decides no radiation, no chemo, nothing, no standard of care, just ketogenic diet and supplements. And I emailed him just a week ago, and he's doing fine. I mean, he's working through the system, but this summer he'll be out 3 years. He didn't have to go to Oregon. You know, the issue here, there are choices. People can make choices when they have the information needed to make the correct choice. Now, if we're going to develop therapies that work and therapies that can resolve and manage this disease, we need good preclinical mouse models or animal models for the disease. And we've built one of the best models that there is that replicates all of the major characteristics that you see in human glioblastoma. That's the VM-3 model, and Christos just mentioned that a moment ago. It replicates all of the major growth characteristics of the human brain, including extraneural metastasis. These tumors arise spontaneously in the mouse that is syngeneic, has a normal immune system, and the genetic background of the tumor and the host is the same. And it even has extraneural metastasis. If you have a good model of glioblastoma, animal model, grow it, take it and grow it outside, and if it spreads throughout the mouse, that's the model that you want. If it grows as a big ball, that's a stem cell tumor. You can kill those easy. The spreading ones are much more difficult. I also want to mention, Pablo's not alone. There are many more people now going down Pablo's route. Andrew Scarborough is here. He's another individual. And I spoke to Andrew, and Andrew's telling me more and more people are choosing the Pablo path. Now, the other thing here is that how these tumor cells rip through the mouse's brain is the same as they ripping through the human brain. They grow across the surface. They grow across myelinated fiber tracts, interfascicular, through the ventricles. Look at this, beautiful tumor cells around the blood vessel, just like I showed you with the human GBM. They're using these blood vessels as railroad tracks to get through the brain among some of these other structures. That's why it's so hard to kill these tumors. Now, of course, I got into this ketogenic diet a while ago. Calorie restriction, ketogenic diet. It was working really well on a lot of tumors. It wasn't working on this to the extent that we thought it would work. Look, this is a mouse that eats all he wants. You put the tumor cells in the brain, and they invade right through. You can see these chunks of tumor well in advance of the major tumor right here. You put it into this brain, and this guy we calorie restricted, and we use calorie restricted ketogenic diets, 2-deoxyglucose, calorie restriction. And my students were saying to me, it's not going down like the other tumors. What's wrong with this tumor? How come this tumor refuses to be beaten down by calorie restricted ketogenic diets? We damn near starved this mouse to death to see if we couldn't stop the tumor. Now, what it did do is it significantly reduced invasion. So the invasive properties of the tumor were markedly reduced, but we weren't killing the majority of the tumor cells. Why are these guys surviving when we're throwing the kitchen sink at them? And we threw temozolomide at this guy too. We couldn't stop it. Do you think it could be the glutamine? So we chose this to test the hypothesis. We used DON, 6-diazo-5-oxoneuroleucine. It's a glutamine analog. It comes in, it blocks the glutaminase activity, you can't take it up, it just gums up the whole glutamine system. It reduces TCA cycle substrate level phosphorylation, which we just heard from Dr. Christopoulos, and it also halts DNA and RNA synthesis. What happens when we treat these tumors now with the ketogenic diet and this glutamine inhibitor? All right, so this is our strategy, this is the way we do things in our lab. We don't put cells in the brain. We put a fragment of a tumor from another mouse into the brain. So that tumor already has a microenvironment ready to go. It's a stage 4 GBM as soon as we put it into the brain. All right, we don't use cells that you start treating with cells, you get good results. You try putting a chunk in there where it's already got a microenvironment, you see what happens. We let it go for 3 days. Now it's a stage 4 here. And then we put them on a fast all the mice, all the diet, and then we give them back either high-carb diet or ketogenic diet restricted, and then we wait a few more days to let this thing, really settle in, and then we use pulsing with DON every other day, either 0.5 or 1.5 mg, and then we use in vivo bioluminescence imaging, ex vivo imaging, take the brain out of the mouse, put it in the dish, and bioluminesce it, because the bioluminescence will pick up the living cells. Now, we said, wow, how come these guys are why do we stop at 15? Because the guys chomping down on the high carbs are dying. They're starting to get morbid at 14, 15 days of age. That's how fast. We already put a stage 4 brain tumor in these mice, all right? You have to realize this. So now we're going to look at the data. These are unpublished observations that I thought the need to share with you, all right? So we use bioluminescence imaging. And here's the guys eating the high carbohydrate standard diet with no treatment. So they're the control for the study. And then using the DON by itself, we got a 44% reduction of the growth of the tumor, the light. So absence of light means you're killing the tumor cells. And I'll show you the evidence. The diet by itself had some minor effect. It stops invasion. But as you can see, we're not slaughtering these tumor cells with the diet alone. We put the diet and the drug, and we got a powerful synergy between the diet and the drug, and some of these mice had no light. So we went back and repeated the experiment, only this time using a higher concentration of DON, just a little bit higher, and using the ketogenic diet restricted now as our control. This is the group that usually mostly represents your experimental and other studies. We're using this as the control compared with the diet and the drug. So we're pressing the system with the diet, and we're pulsing the system with the DON. And what we found is that this is background light, almost no light. Only one guy had a significant amount of light in the treated group. And it turned out this was the one mouse that was looking a little finicky, and we said, let's not inject him today. So we didn't inject him, and he was the only guy that had a little bit of light left in his with missing one injection. So we do the average of this, and we have massive, unbelievable amount of death and reduction when we targeted both the glucose with the ketones and the DON as a pulse. And this is what it looks like when you put it in the Petri dish. So we take the brain out of the mouse, you can see it here, and then we do the bioluminescence imaging. Standard high-carb diets, ripping, okay? They're out of the program in 14, 15 days. The ketogenic diet, guys, yeah, you have a lot of light. I told you, the ketogenic diet alone is not going to resolve these GBM, at least this one. And the fact that humans do so much better than the mice is blowing my mind. You know, humans do much better than the mice. And then if you put the two together, the restricted diet with DON, we get no light. What does that mean, no light? Does that mean there's no GBM? Does that mean it's resolved? So we decided, let's do the histology on this thing and see what's going on inside the brains of these guys. So H&E staining, a pathological necessity. If you're going to prove anything in neuropathology, you have to do H&E staining. So what you have here is you have standard diet, unrestricted. Look at the mass of the tumor, and you can see the tumor cells migrating, invading, I should say, into this white. This is the invasion. And that's under low power. Under high power, these cells are so healthy. These cells are growing, they're packed, they're piling on top of each other, massive cell density, mitotic figures. This is like what you see in human GBM. Now look at, here's the ketogenic diet restricted. There's a sharper demarcation. Again, the invasion is slowed down, and there's more spacing between the cells. But the cells are not dying. Now there's some that are, but basically they're alive. So then we put the diet and the DON together, and we got massive death. As my associate, Purna Mukherjee said, you got a battlefield here. This is slaughter at the highest level. These are not live cells, these are fragments of cells. All right, and this is consistent with the fact that there's no light. I don't know if we cured this mouse. We didn't do the full studies. I don't know if we resolved this mouse completely. All I know is there's no light, and we got massive death. And here's another very important and very interesting observation from this study. It turns out that when you give the DON, here's the amount you get into the tumor in the brain on the high-carb diet. But with ketogenic diet, we got almost twice as much DON. Drug on target is the ketogenic diet is. We saw this a long time ago for another drug. All right, it was a drug that we were using for treating ganglioside storage disease, and when we gave it under a ketogenic diet, we delivered 3 times more drug to the brain. So I'm not really sure what's going on here mechanistically, but it's very interesting. And then this is the work from Angela and Dominic. And as I said, if you have a good model of glioblastoma and you put it in the flank, it should spread throughout the mouse, and that's exactly what we're seeing here. Labeled tumor cells implanted subcutaneously, and they were using now hyperbaric oxygen as the pulse and the diet as the press. And the oxygen, you know, had a little effect. The diet, of course, has a little bit more effect, but you're not resolving. But when you put the two together, you get synergy. Press, pulse. Now, this is we have this figure, Dominic and I, we have a paper that we've submitted it's under review, but we built this figure as an example of the strategy that we plan to use to manage and hopefully resolve cancer. What I did was use Da Vinci's Vitruvian man symbol, what we did. So we have an individual who has metastatic cancer, and we're going to try to manage the metastatic cancer, and then we're going to try to resolve the metastatic cancer, whether that's colon, whether it's breast, whether it's brain, bladder, whatever. The strategy will work because all the tumor cells are the same disease. Cancer is a singular disease of energy metabolism, thriving on fermentation fuels. So the way to manage the disease, you have to press and you have to pulse. Press therapies can involve, but not exclusively, restricted ketogenic diets, and sometimes you don't need to restrict the diet, and you can have various combinations of the diet. Paleo-ketogenic diets. Some people respond better to one type than another. Fat composition of the diet plays an important role, so you need to know that. Ketone supplements can be very important as a continual press on the system. Stress management is extremely important if we're going to resolve cancer. People get emotionally freaked out when they have cancer. Believe me, they get depression, they get all kinds of things. If you're not able to deal with their emotional stress, it's not going to resolve as quickly as it could. And we use, what is this? We use exercise therapy, yoga therapy if necessary, music therapy. There's a variety of different therapies that actually can reduce physiological stress, and that we keep on the patient so they feel good all the time. You want to keep their spirits up. You want to keep them involved in the management of their disease. And then while this is going on, we then institute pulse therapies, which is glucose inhibition. We can take, part of that is coming from the diet, but we can boost that up a little bit when we use glucose inhibitors, 2-deoxyglucose, fluorodeoxy. There's a whole range of drugs that could be applied here. Then we have glutamine inhibitors. I just gave you an example of Don, okay? Because we were just trying to test hypothesis, just to test the hypothesis. So there might be other glutamine inhibitors that could work as well. And then we have hyperbaric oxygen. So we have, these are all press or pulse therapies. Now, the strategy is called dosing, timing, and scheduling. How much doses do we use? What is the dosage? When and for how long do we do this? And what is the schedule? All right, which comes first? This one, that one? How do we schedule this? And in the intermediate periods, we can add back certain therapies to enhance the health and vitality of the person. So what we're going to do is use this. This is a new novel therapeutic strategy for the metabolic management of cancer. And it is my opinion that this, in time, will eventually make most other standard therapies for cancer obsolete. Because this is based on the biology of the system, on the biochemistry of the system, and not some genetic nonsense of the system. Okay? I'd like to thank the collaborators that have been working with me on this. And I'd especially like to thank the Single Cause, Single Cure Foundation that Travis Christofferson has set up. It's a beacon. He's one of the few guys that really understands the nature of the problem. And people like Ed Miller who support this is very important. George Yu Foundation. These people seem to understand the nature of the problem. I don't know why these few people understand. There should be a lot more other people understanding what the problem is. It's almost like there's some sort of a wall to prevent people from understanding what I just told you. And of course, my own research. And thank you for your attention.